the purpose of today is to go through a series of case studies. So we've had an introduction to glycans and carbohydrates, mucins with GLEB, and today we're going to look at specific case studies. The first of these is gastrointestinal mucosa in health and disease, building on from what Gleb's been talking about. Then Gleb's going to follow up with your case study on
dietary fiber, mucus adhesion first, and then dietary fiber.
Then I follow up with a third case study, which is glycovaccines against meningitis and all this sort of stuff, which leads beautifully into Tony's final
Case study,
which is the COVID glycans and vaccination. And then on Friday, we've got Linda Bellick-Malin coming in, which on cellulose derivatives. She does a super show. And then in the afternoon on Friday, we've got Immo Friedrich, who's beaming in from München, from Munich in Germany. Immo's coming on Friday, Tony.
Oh, that's great. That's great.
Yeah. But we're going to kick off now with Tony. Gleb, does you want to say something?
No, I think all being said.
Before you start, Tony, two things. What time have you got to be at your BLT class? Off what? Oh, we'll finish at 25 past. No, we'll finish well in time for lunch and things. And then on Friday, we've got a little do. If you're around on Friday evening, we'll tell you more about that later.
Fantastic. Yeah.
So, Tony, over to you.
Okay. Good morning.
Oh, that's fine.
Yeah. Good morning. A rainy occasion. Can you see the screen? Can you see the talk?
Yes, we can.
Yeah, you can. Okay.
Before you start, Tony, just to say that me and Tony go back, what, 1,000 years?
Yeah, probably more.
We were both postdocs at the University of Bristol working on...
and you've abandoned that ship under the pub in Wirral.
Oh,
that's you abandoned that ship. That was yours, right?
No, it was my ship, yes. They weren't in, so we left it on the car park, yeah. But Tony was there when I was working with Michael Creeth. Where are you, Tony? He was in the next lab. This is back in the 1980s. I won't tell him the lab coat story.
No. Save that for a rainy day. Well, it's a rainy day, I guess.
Right. Okay. I'm back in 5 minutes. I'm just going to get a charger, but I'll leave you.
Okay. Right. Okay. Then I'll go ahead. So what I'm going to be talking about today is the gastrointestinal mucosa in health and disease. And this is pretty much an overview of what's going on, and focus on a couple of the things that we've actually been doing over the years. So that'll include the mucins, the trefoil factor family peptides, and glycobiology in general. I should mention that the glycobiology, the background to that is going to be in more detail in the second talk. But there's plenty of backup on this, and this talk and the other PowerPoint presentation will be available. So you can pick that up and browse it at your leisure as you like. Okay. So let's go. So, so basically, as I said, there's a literature list available, and if you want more background information, there is the Essentials of Glycobiology, which is available at the website, and it's free. So there's a lot more glycobiology that you can obtain if you go to this website. So outline of the talk. It's lost a couple of things here. Let's see. Oh, right. Okay. So what I'm going to cover is, first of all, the human gastrointestinal barrier itself, and the mucins, trefoil peptides, and glycobiology. Move on to colorectal cancer, the adenocarcinoma sequence. Move on to the inflammatory bowel diseases, ulcerative colitis and Crohn's disease. an example of a disease in the pediatric gut necrotizing colitis, and then models of disease using the MUC2 knockout mouse and cultured colorectal cell lines. So first of all, the human gastrointestinal barrier. And here we have the mucin gene family, which has 22 known genes which belong to this family. That's at the current time. And there are two main types: the secretory, which are secreted out of the cell, and most of them will actually form gels as a protective layer, and the membrane bound type. And each gene has a characteristic protein domain structure, which is characteristic for these for the mucins. In addition, there is extensive glycosylation on their tandem repeat domains. I'm having a problem with this this little bar across the screen. It's hiding some of the things that I can see here, but I'll try and push on. Okay, so here are schematic cartoons of two typical types of the main mucins that we find. First of all, at the top, the MUC2, and you can see that in the central VNTR, variable number tandem repeat domains, that's where the sugars are. You can see the glycosylation. On the bottom, you can see MUC1, which is a typical membrane-bound, membrane-associated mucin. And you can see there are two domains, the SEA domain and another domain, which are the links through and onto the to the membrane. We can get signaling through here as well, and then the VNTR is up this end, and this extends outside the cells, and the glycans we find are associated with this particular region. Here is just an to give you an example of the sort of peptide domains that we find in the mucins. At the top, you can see the different mucin genes listed down on the right-hand side. On the top are the secreted mucins. So MUC6 and MUC2 is one that I'm going to be talking about a lot. So that is there. And you can see the membrane mucins. MUC1 is down here. 12, 13, 3A, and 3B are all membrane-associated mucins. You can see that the blue color, this tandem repeat VNTR region, is common to all of the mucins. In the secreted mucins, there is a cysteine-rich domain. You can see this with the brown here, depending which mucin, secreted mucin we're looking at. And for the membrane-associated mucins, you can see these black domains here, which are the membrane linking domains. So characteristic properties that we see. Here's just a schematic to show you what these mucins actually do. Here are the epithelial cells, whether it's the GI tract, respiratory tract, or whatever. And we can see that the mucins, the membrane mucins, are here in the glycocalyx, attached to the membranes. The secreted mucus, which may be gel forming for MUC2, for example, is secreted and forms a layer in the lumen protecting the surface of the mucosal cells themselves. Here is an immunohistochemical slide showing the presence of MUC2 in the human colorectum, and you can see the goblet cells in these crypts are packed full of the MUC2 mucin. If we go to immunohistochemistry and we look at these crypts again, on the left-hand side you can see a crypt which stains light blue and the dark blue black color. So that's a mixture of acidic radicals, the blue for sialic acids and the black for sulfate. Here is a crypt which is largely sulfated, and here is one which is rich in sialic acid, so it's largely the lighter blue color. In addition, the mucus gels contain protective proteins, and there's a list here showing all of those that that have been identified: defensins, lysozyme, transferrin, secretory IgA, gastrokines, and the truffle factor family peptides, which I'm going to tell you more about now. So I'm giving you one example of these protective proteins as an example. These peptides, known as TFF peptides, are a group of 3 peptides, which are small. They have only 55 to 105 amino acids. They are abundant in the gastrointestinal mucosa, and they bind to mucins. TFF1 binds to MUC5AC, the gastric surface, TFF2 to MUC6 in the stomach, and TFF3 to MUC2. So, as these peptides are highly expressed in mucus-secreting mucosa, they have a role in epithelial protection. They interact with the mucins to help gel formation, and in restitution. What that means is that if damage occurs to the cells in the epithelial layer, in the gastrointestinal tract, the trephine peptides trigger pathways which allow the cells to grow back effectively and repair any damage that occurs at the mucosal surface. Now, here's a schematic again showing you the 3 main TFF peptides: TFF1, 2, and 3. TFF2 in human is unusual because it actually forms a dimer, but you don't need to worry about that. We're going to be focusing largely on TFF3, and here, just to demonstrate that we can detect these quite readily in the normal human colorectum. On the left-hand side, you can see the immunohistochemistry staining for TFF3, and on the right-hand side, in situ hybridization, and again we can see abundant staining for the TFF3 nucleic acid. So let's move on to the disease side of things. I'm going to talk a little bit about colorectal cancer. Here is a resected section of the colon, and you can see at the top there are polyps from this patient, and at the bottom you can see a typical adenocarcinoma. So this is the, if you like, the in situ picture that you will see. Now, what is important is that there is a sequence of events leading up to the carcinoma. So if you look over on the left-hand side here is the normal epithelium. Go across to the red box, which is the carcinomas, and metastases come, spread of disease after the carcinoma has been formed. But in the middle, you can see there are a number of different states, which are basically called adenoma, and this is a progression. As you go from the normal epithelium through to the late adenomas and on to the carcinoma, there are a sequence of molecular events, gene events that occur in the mucosa, which lead to the changes which are typical for this disease. So we can start to screen for this. This is important because if we'll be talking about inflammatory bowel disease in a little while, and the inflammatory bowel disease don't reach the carcinoma stage. So they are characteristic for these different stages as we go through this adenoma carcinoma sequence. So what about the mucins in colorectal cancer? we know that the adenomas show a change in the subcellular distribution of MUC2, but there are limited changes in the expression level, so we don't get more or less. The carcinomas show a change which is based on their phenotype. Downgrading of MUC2 expression and translated protein only occurs in colorectal tumors with poor differentiation. And changes in the tumors are preceded by the sequence of events that I've just described. So let's look at the inflammatory bowel diseases. There are two main types that have been identified: ulcerative colitis and Crohn's disease, and these vary a little. So first of all, as I mentioned just now, important to remember that this is a precancerous condition, and it leads to changes in the mucus layer in the gut. So what we see is a reduced thickness in the adherent mucus layer in ulcerative colitis, but an increase in Crohn's disease. So there is a difference between these two IBDs at that level. We find reduced turnover in ulcerative colitis, but not in CD. And there are no changes in the type of mucins expressed. MUC2 is maintained. And this is different to the carcinoma, adenocarcinoma type, where we see the induction of other types of mucins. And again, that's another big story which I won't go into. Some of the changes that we see here are also mirrored in necrotizing enterocolitis, as we'll see a little later in the talk. And here's a bit of information about the expression of the secreted mucins in ulcerative colitis. So if we look down the left-hand side, we can see MUC2, which is the main mucin in the colorectum. We don't see any MUC5AC, which does get switched on in the adenocarcinomas. There's no 5B and so on down here. Nothing at all here. And if we look at the patients that have been studied here, on the left-hand side, first of all, with the this is immunohistochemistry and in situ hybridization analysis, scored as pluses. Here's the normal pattern that we see. Here are all of the UC colitis patients together, and then the mild, moderate, and severe disease. And we would expect this to be nearest to the carcinoma, adenocarcinoma state. So if we're going to see any changes, we'd expect to see them at this stage. And if anything, MUC2 doesn't change very much at all across this this range of diseases. Perhaps we would expect to see if anything is turned up at all. What we do see is a change in the glycobiology. And here is an example of sulfo-Lewis A. This is a structure. I'll show you the structure in just a second. Remember, this is in ulcerative colitis. What we do see, if we stain for this glycan that is attached to the mucins in the colorectum, here's a normal pattern. Here's mild disease. There's not much change. You can see the crypt quite nicely, but there is a reduction in the staining in severe disease. So as we progress through ulcerative colitis to severe disease, we see a change which relates to a glycan structure which is carried by the mucins. Here are the structures of these glycans. Now, if you imagine, and again, you'll see more detail about glycan structures in the second talk, so you confer across to that later on. Sulfo-Lewis A down at the bottom is the normal structure that we would find in abundance in the colorectum. Sylar Lewis A and X, which we can see on the top, the structures are related. The sulfo Lewis X has a negative charge due to the sulfate, in contrast to the sialic acids, which you can see in the Sylar Lewis A and X structures here. So again, this underlines the importance of screening for glycan structures if we're looking at disease in the gastrointestinal tract. If we look at the membrane mucins in ulcerative colitis, again, we see very little change except for MUC1. Again, here we've got the same, here is normal, here are the colitis patients, here is severe ulcerative colitis. And if we look here, we can see that there is an increase in the staining for MUC1 in severe ulcerative colitis. There is very little change in any of the other mucin genes that we see. Let's switch over to the trephol peptides in ulcerative colitis. What do we see here? What we find is a loss of TFF3, which is normally expressed, as I showed you before, and an upregulation of TFF1, which is at lower levels in the colorectum under normal conditions. These changes for TFF1 and 3 are important in IBD because they link through to the ERK MAPK and the NF-kappa B regulatory pathways which operate in the colorectum. So here is an indication that this is an important regulatory link associated with the disease. And here is the data that we have for this. TFF1, 2, and 3. And if we look across here, we can see an increase in severe disease for TFF1 and a downregulation of TFF3 in the severe group. There's not much change in the mild or moderate. So this mirrors the changes that we were seeing for the mucins in ulcerative colitis. And again, here, immunohistochemistry, here for the loss of TFF3, we can see on the right-hand side much weaker staining for TFF3 in this tissue section from a colitis patient. So let's move on to pediatric disease. Clearly, there are a whole range of different conditions that we could study. This is one that we chose to study in Bristol, and is a disease of the pediatric gut. So this is for very young children. These are literally children who are just a few days old. The pathogenesis is still poorly understood. We have some ideas, but there's still much to be worked out. And there are a number of potential triggers that may lead to this disease that exist in a susceptible host. Prematurity, in other words, children that are born before the normal time. This is a major risk factor, and the studies that we've carried out, it may be that an immature supramucosal barrier also plays a role. NEC, necrotizing enterocolitis, is characterized by a failure of intestinal protection, and obviously this is a crucial situation for newborn children. The children show a dramatic loss of a mucosal epithelium. And the microflora is implicated in this change, but the etiology is not determined. Obviously, this is a very difficult situation to study because these are children who are very, very young indeed. So what can we actually demonstrate? If we look in the normal fetal gut, so this is gut just prior to birth, we can see normal staining for MUC2. So that is as it should be. As it should be. If we look to see how these cells are turned over, we can look at Ki67, which is a cell cycle regulated nuclear protein, which is expressed by cells at all stages of the cell cycle and is used to screen for proliferation. How are the cells growing? So here's some immunohistochemistry. Over on the left-hand side, we can see the pattern that we'd see in the normal gut, and you can see the Ki67 staining at the bottom of the crypts, and same in the colon on the bottom. In necrotizing enterocolysis, we can see that this pattern is not only reduced, but it is more sporadic. Now, this is just a high resolution section for the staining that actually takes place in the bottom here. And in the recovering patients, the recovering children, we can see this is coming back towards the structure that staining pattern that we see in the normal situation. Again, at a higher resolution on the bottom, on the right-hand side. Here is the evidence for the loss of the trifosfatide TFF3 in necrotizing enterocolitis. The top two slides show staining in normal cells, and we can see here at higher resolution there's strong staining in the goblet cells, so very clearly. And in the bottom, again, at two different magnifications, we can see that there is a significant loss of TFF3 expression, and we can see some of the goblet cells which are empty. There is no staining at all. If you compare the bottom right with the one directly above it, you can see there's a dramatic loss in this disease. So to sum up what is going on here, there's a massive loss of epithelium expressing TFF3 and with proliferative potential with the Ki67. So the dramatic epithelial faults that are occurring in NEC. The decrease that we see for the mucosal TFF3 means that the protective and restitutive recovery responses are diminished. So these children have much poorer defense as a result. And although we see an increase in TFF1 expression and to some degree proliferation, this isn't sufficient to reverse the damage which is done by the onset of NEC in these children. So let's move on to the final stage here, the models of gastrointestinal disease. Obviously, it would be nice to set up systems where we can actually look at the details of the disease in situations that we can use experimentally. And there are two examples that I'm going to give here. First, the MUC2 knockout mouse, and second, colorectal cancer cell lines, and I'll explain what these are. So we can engineer the MUC2 gene in order to delete it. This is the detail of that. Clones were isolated, and the progeny with the plus-minus progeny back-crossed to give all of the expected outcomes, including the MUC2 minus, which is the knockout mouse itself. So, what are the properties? First of all, the MUC2 minus genotype is a null mouse. Secondly, no other mucin genes could be detected. So there was no MUC5AC, MUC3, or MUC13 that could be detected in these cells, in these mice. The proliferation and migration rates in these mice in the GI tract were increased in the MUC2 minus. Apoptosis was decreased, and I can't read the bottom line there. There's the bar is over the top. Anyway, let's move on. The experimentation which was carried out in order to screen for these changes was looking for MUC2 mRNA and protein, the presence of goblet cell mucin stained by the Alcium blue periodic acid Schiff stain, which is a classical histochemical stain, the effect on a co-localized protein, and again, the trephol peptides were used here, so the expression of TFF3 was screened. Proliferation was checked out using the standard bromdeoxyuridine assay. Cells in the S phase were screened. Apoptosis was measured using the tunnel assay. And again, I can't read the bottom across there, but you should be able to. Oh, that's the bromdeoxyuridine assay. All of these are standard techniques which can be easily carried out in the lab. And the results that came back were, first of all, epithelial tumors were found representing multiple stages of tumor progression. You remember back to the adenoma carcinoma sequence that I mentioned before. Young mice showed small intestinal adenomas. So these are before we get to carcinomas. The older mice showed a majority of invasive carcinomas. So this again mirrors the changes that we saw in the adenoma carcinoma sequence. And there was no influence of inflammation in these in mice at all. So here are the data. At the top, we can see on the left-hand side here, we can see the MUC2++ mice. So these are effectively the normal mouse, looking at 6 months and 12 months for the number of tumors, the gastrointestinal tumors. And we can see there is nothing in the normal, excuse me, nothing in the normal mouse, but we can see the GI tumors appearing at 6 months already in the mice, and the invasive tumors in the older mice. So, what can we conclude from these studies with these knockout mice? First of all, MUC2 plays a role in intestinal homeostasis. We need MUC2 if the mucosa is to function normally. The absence of MUC2 induces increased proliferation, decreased apoptosis, and increased migration. So this looks as though there is a cellular response to try and recover the damage done by the loss of the MUC2. So we can assume that the lack of mucosal protection and lubrication, because the MUC2 will also form gels on the surface and give the mucosa its normal protection, but also with MUC2 signaling through the protein is affected. And there is a spontaneous progression to invasive carcinoma without any inflammation taking place. So these are all characteristics which match well with the human samples that were taken before. So this is a useful model of the disease. If we move on to the second model, we can use cell culture of gastrointestinal cells to look at the adenoma carcinoma sequence. The way that this has been done is that if you look at the top here, a benign adenoma was isolated from a patient, and cells were extracted from this adenoma. They were then cultured through and taken through different culture regimes, and they developed into two different types of cell lines, which represent on the left-hand side the PCAA94, which is shows characteristics of a mucinous type carcinoma. Again, one of the types of carcinoma that we find in man. On the right-hand side, we can see the PCAA SB10, which is typical for an adenocarcinoma. We saw the adenocarcinoma in the resected section earlier on in the talk. So we have two cell lines, the PCA94 and the PCA SB10, which are typical for two different types of cancers which we find in man. And because it's a cell culture system, we can manipulate this to look at the different patterns of expression of molecules that are in these cells. Here's a micrograph just to show you. This is PCAA10 cell line, and we can see the typical cells that we would expect to see in the colorectum. There's an absorptive cell over here on the bottom on the left side, and at the top we can see typical goblet type cells which are full of vesicles, which where we'd normally expect to see the MUC2 prior to secretion. Now, in order to study these in more detail, we wanted to look at mature and non-glycosylated forms of MUC2. This will give us an idea of the biosynthetic progression which is occurring in the cell lines that we want to study. And we used two antibodies in this study. If you remember right back at the beginning, I showed you a picture of MUC2 in the normal colorectum, and this is the antibody that we used for it, the LUM23 antibody. This locates to a peptide sequence which is in the mature mucin, which was away from the glycosylated regions in the variable number tandem repeats. You can see those shown in red here. So this antibody will detect mature MUC2 mucin. Now, in contrast, we have another antibody which has been prepared, the 4F1 antibody, which binds to a peptide sequence in the tandem repeats. So this is an anti-peptide antibody. Now, this is important because it means that we will only detect these sequences here, provided there are no glycans present. So without glycosylation having taken place, as we see in the mature molecule at the top. So we have two tools that we can look at the progression of the biosynthesis in the mucins in these colorectal cell lines. So the first thing that we did was to look at the cells. So these are the PCAA 10 cells. What we see is that in the cell layer extract, we can see, and this is a fractionation on cefacryl S10, S1000. So we're looking at size fractionation here. And we can see in the cells, as we would expect, a high molecular weight peak at the front, and a lot of other material in the cell. In the in the cell layer extract at lower molecular weight. In the medium, we see essentially just the high molecular weight mucin peak, and these are the staining with the LUM2-3 material is essentially in this initial peak, high molecular weight peak here, which is present in the medium. The 4F1, the precursors, we see only in this lower molecular weight peak in the cell layer extract, and this is as we would expect. If we put these onto density gradient centrifugation, at the top we can see screening with the proteoic acid shift stain and at 280 nanometers, and we can see a typical mucin-related peak. This is where we would expect to see mucins on the density gradients. And these are Western blots at the bottom showing that the 4F1, the precursors, as we would expect, are present at lower molecular weight in this region here, whereas the LUM2-3 staining matches up to the high molecular weight, higher molecular weight mucin peak that we see in the density gradient. So that's 4F1 and the LUM2-3. Now, if we take the PCAA medium, and you remember this is the adenocarcinoma cell line. Again, if we look at the staining here, we see a big peak on the density gradient in the area where we'd expect to see the mucins. And if we look for the staining with the two antibodies, we find there's only reactivity with the LUM2-3 for the mature mucin, and there is no 4F1 detected at all. And this is what we would expect in the secreted material. This is in the medium. If we switch now to the PCAA 94 cells, this is the mucinous carcinoma. Here, the pattern again, on the density gradient, we see a big peak here where we'd expect to see the mucins with the PAS and 280 nanometer stain. If we look for using the antibodies, we find that this matches up to the mucin peak as we would expect, but relatively low reactivity with the 4F1. So if we go to the staining with these different mucins and their precursors in the cell mucins. These are what we'd expect to find inside the cell. We see the LUM2-3 mature mucin as we would expect in the higher molecular weight range on the gradient, and the 4F1 staining in the precursor stage where we saw in the other cell line as well. If we now go to the medium, we see we have a higher molecular weight mucin type peak for the staining of LUM2-3 and with the PA stain, but there's another peak here which shows up, and the 4F1 doesn't show a lot of reactivity in the assays there. But if we go to the staining on the Western blots, then we see that in the medium, so this is secreted material coming out of these cells, we see the staining in this high molecular weight mucin peak as we would expect. But the 4F1 shows that in the medium, the secreted material, there are a whole range of precursor molecules which haven't been fully glycosylated and which are being secreted as well. So this BCAA94 cell line, mucinous carcinoma cell line, shows an abnormality which relates to the biosynthesis of MUC2 in these cells. So to sum up these results, the PCAA10 cells show morphological similarities to adenocarcinoma and secrete mature MUC2 into the medium. So that is as we would expect. In contrast, the MUC2, the PCAA94 show mucinous carcinoma pathology, but secrete both mature and precursor forms of MUC2 into the medium. And that is a difference that we can look at perhaps in more detail in further experiments. So what did the talk cover? These are the points that we looked at. The gastrointestinal barrier with the mucins, trephalpeptides, and glycobiology. Colorectal cancer with its adenoma carcinoma sequence. Inflammatory bowel diseases, changes that occur in ulcerative colitis and Crohn's disease, pediatric gut necrotizing enterocolitis, and 2 models of colorectal cancer that we covered. Okay, so as I said at the start, this talk was based on my own areas of interest in research, as you probably picked up. There is a literature list available for the talk, and that covers most of the points that have been raised here, plus a few extras as well. If you have any questions about the talk, the easiest thing is to send me an email. My email is here and also on the literature list. Obviously, there's a huge amount of information here that you can try and digest. But certainly, please send me an email if you've got any queries, and I'll do my best to answer it.